tough time because neurophysiologists simply don't believe this information. So he was one of the pioneers in the study of glial cells. He started his career as a bachelor's in the University of Minnesota and did his master and PhD degrees in the University of Washington in St. Louis, Missouri, which is one of the most prestigious universities for the medicine area in the United States. And after that, he was really interested in research. And I think the talk of that will give you an impression because this is one of the areas that he has been really, really interested as a neurologist. And that's why I think, if he will correct me if I'm wrong, that's why he became so interested in glial cell physiology. And he was trained later on in Stanford University. And at that time, he was really, really hanging out with a golden age. I mean, we have discussed that. Most of these guys just having fun, as Bruce told me one time, just hanging out, going for beer, and doing extremely nice science. That was a golden age because these guys were just publishing Nature, Science, and most of these guys like Krickstein, if I'm not, it's one of the leading researchers in neurogenesis, for example. So he was able to interact with these guys in Stanford University. Later on, he went to Yale University and did some physiology with oligodendrocytes and white matter. And as you can see, these names are not low, cannot be mispassed. Stanford, Yale, and then he went to Berlin, did some work with Professor Ketterman. He's also collaborating with Mike Nedergaard in Rochester University. I mean, I cannot finish to tell you all the things that Bruce has done, but most importantly, he keeps doing these things. It's not that we talk about the past. We can talk about what he's doing right now, and it's really, really inspiring for the young people and for all people like me, because the energy that this guy has is just impressive. And today he will talk us about not petrified wood, Bruce, but about white matter and metabolism in the brain. So I don't continue, just let you listen to Bruce, and hopefully you can interact with him, ask questions. He's very open, so he will be glad to answer any questions you have.
Daniel y Octavio, muchas gracias por todo. Lo siento mucho, hablo inglés solo, not even Spanglish. So I am going to take the liberty of making a few comments, and I don't know if the audience can see well from the back. There was a light, Daniel, that might make it a little easier. So I thought I would take a chance and just have a brief philosophical conversation with you about glial research. Glial research is hard. The cells don't identify themselves easily. Daniel was just giving us a talk. He used the comment silent cell. When you put an electrode in them, they don't make any noise. So from the very beginning, it has been challenging to study glial cells. It's gotten easier. There are many wonderful tools that we can employ today that we didn't used to have, that you'll be able to use and appreciate and be successful with. But the field itself is difficult, and there's no getting around that. There's been a huge amount of speculation. People that do have it 30, 40, 50 years ago, talking about how glial cells were going to be the answer for memory and learning. They were going to be the answer for every disease process. We made wild overstatements that we've never been able to really deliver on. And I would like to recommend that as a group, all of us try to avoid that. From this point on, I think it makes more sense to really insist on rigorous demonstration of the facts. And if we do that, we're going to find that more people flock to the field of glial research. And most importantly, for everyone in this room, I think, is that funding agencies will be unable to argue with us as we bring more and more good ideas and convincing proof. They're going to say, here, take this money, go do good research. There are going to be some difficulties. We're not out of the woods, even though things are much better today in terms of glial research. It's going to certainly be true that we're going to face the business of differences in different glial cell types. No talk given in the last 3 days has failed to mention that there are many different types of glial cells. And it may even be that under one circumstance, the glial cell behaves in a particular fashion and in another, yet another fashion. So we're going to have to face up to the reality that there is not just one astrocyte. That may be upsetting. It certainly was to me when I first thought about it. It's a lot easier to study rodents than to do intracellular recordings from human beings. But if there are any volunteers in the audience, I'd like to take your name down. And I'll buy you a plane ticket if I can record from your glial cells. No? No takers. All right. So we're stuck using animal models, but the real reason that we do science is not to understand what a mouse astrocyte does, but it's to understand what your and my astrocytes do. We need to be looking for opportunities of extending our observations, testing our observations in human preparations. They may come from a surgical specimen. They may come from immortalized cell lines. They may come from a small bit of tissue that's harvested when a surgeon has to remove a tumor. There could be lots of different ways, but frankly, that's on the horizon. It's unavoidable, and I think it offers brand new opportunities for understanding how the human brain works. I mentioned already that we can't do anything without money. And every single person in this room is an ambassador. You are a spokesperson for science. When you're having a beer, a cerveza with your friends, you should be talking about how important science is, about how it leads to discoveries that lead to improved living, conquering diseases, understanding better ways to learn. If we don't do that, no one will do it for us. And if we don't do that, eventually people are going to stop being interested in funding our work. So the value proposition of science needs to be defended, and the people that are in the best position to do it are all of you. So Daniel mentioned the golden age. I actually think the golden age is now, Dan. I don't think I lived in the golden age, maybe just the tail end of it or the very beginning of it. I think you're seeing the golden age. We know so much right now. Everybody that's influential in neuroscience is finally making the admission, standing tall and saying plainly that we have to understand glial cells if we want to understand the brain. So we are in a spectacular position. And join with me. I'm starting a small political movement. Let's not call them support cells. That makes them a lowly type of cell that's simply supporting the only cells that really matter, the neurons. Let's call them partner cells. I'm willing to be partners with neurons, but I don't want to support them. I don't want to serve them. Now, I think most of the audience are probably students. Can I have the students in the audience raise their hand? If you're a graduate student, if you're a PhD student, if you're a postdoc. All right, I think I'm right. So, you know, being a graduate student is not a bad thing. Here's a graduate student from an earlier age, not the golden age. These were the Stone Age type graduate students. But I put this picture up simply to tell you that the people you work with have wonderful ideas and they're setting a direction for you. They're helping you learn how to master the techniques that you're going to need for science. They give you direction. But it's been my experience that some of the very best ideas come from graduate students. They come from young people who are being taught what today's dogma is, and you just simply don't believe it. Now, you're too polite, right, to say that's junk, I don't believe it. But your mind is sort of saying, that just doesn't make any sense. That's exactly what should be going on. So listen to that little inner voice that says, that's probably not right. I want to try something else, and just go in that direction. If you can dream it, begin it. There's real power there. I'm going to talk about glial glycogen and brain energy metabolism, and the points I'm going to make are the following. Glial cells are the only cells, actually astrocytes are the only cells in the central nervous system that have glycogen. Central nervous system glycogen, I think now has been demonstrated to be very important to neighboring neural elements, and I'm mainly going to talk about white matter, so the neural element that we'll be talking about is the axon. And the way this works is that the astrocytic glycogen breaks down to lactate, which is transported across the extracellular space to the neural element and supports metabolism. I'm going to demonstrate, or I'm going to try to convince you that we have good evidence for this taking place in the instance where there's no glucose whatsoever available, and also in the instance when there's intense neural activity. A very recent story, and this will be the end of my talk, we'll talk about the peripheral nervous system. And it was a big surprise to me to discover that the glial cells in the peripheral nervous system, Schwann cells, also have lots of glycogen, and it is very important to neighboring axons as well. Science today gets done by a cast of characters, and I've listed just a few of the people that I've worked with relevant to some of the things I'm going to talk about today. I don't have time to go down the list and define what everyone did, and in fact, I will offer that as a disclaimer. I feel a little guilty giving a summary talk and not making it clear which student did what, or which collaborator did what part of which experiment, but I'm going to gloss over that today. The other disclaimer is that this is now a large field. It's controversial, and I won't have time today to bring up all the areas of controversy. So when you listen to my talk, feel free to investigate some of these ideas yourself, and what you'll encounter is that there's some variability of ideas. So my outline, very simply, I'm going to make a few introductory comments, and then I'm going to tell two stories, one in the central nervous system about glial glycogen, mainly astrocytic glycogen, and one in the peripheral nervous system, which I will get to at the very end. I'm hoping that everybody in the audience realizes that in the end, the brain doesn't work if it doesn't have ATP. So you have to have ATP to operate the brain, which is a very expensive electrical device. It needs to make and use ion gradients for all of its electrical activity. It turns out that the mouse needs more than you do even, but in human beings, the brain is a high consumer of glucose. That glucose is used primarily to pump ions back and forth and maintain certain transmembrane ion gradients that are essential for excitability. We don't talk about it often enough, but another big use of ATP is the constant reconstruction that takes place in processes in the brain. Both neuronal processes and glial processes are constantly building and breaking down. That doesn't come free. We have to use ATP for that. So that's the way it is. You need all of this energy for this energy metabolism. And you would think if you're that dependent on energy in the brain, surely you would have been smart enough to design an organism, a machine that had some spare energy substrate for times when there wasn't quite enough around. If you had read the manual that came with your brain, and this is my version right here, you would have seen that number 3 states clearly, do not unplug your brain. You've got to have continuous oxygen and glucose, because if you don't, bad things will happen, and it won't take very long for that to happen. If anybody disputes this particular comment, we can go outside later today, and I'll put your head underwater for about 5 minutes, and we'll see how you do. You can hold your breath and whatnot. But so yes, you desperately need that, and in fact, your brain does have glycogen, even though it has been almost completely ignored for the first 150 years of serious neuroscience. Really, it's only been within the last 10 or 15 years that anyone has bothered to take this topic seriously. Let's start with the fact that glycogen is a beautiful molecule, absolutely beautiful. I couldn't begin to show you what it looks like in a true molecular photograph, because I just don't know how to do that. But it's a branching tree structure with secondary, tertiary, quaternary domains of glycogen branches. All of these are glucose molecules, and a single glycogen molecule can have 20,000 to 50,000 glucose units. There's a huge advantage to that. I can take and reduce the osmotic pressure of 50,000 glucose molecules to 1 molecule worth of osmotic pressure by combining it into a glycogen molecule. Very impressive, very clever bit of evolution here. In addition, even faster than glucose, you can get ATP from the glucose that's broken off of glycogen. So for speed of use and for efficient storage, there's never been a thing like glycogen, and certainly evolution hasn't thought up anything better. What you do with glycogen varies from one organ to the next. There's plenty of glycogen in liver, muscle. We'll talk about the amounts in brain in just a moment. In the liver, the glycogen is strictly for export, and you have an enzyme that splits off the phosphate group and allows the glycogen to be imported into your bloodstream. You have enough glucose in your liver. Some have more than others, but you have enough in your liver to last an entire day with no glucose coming from anywhere else or even burning any of your fat molecules. So lots of glycogen in liver for export to the rest of the body. Muscle uses it strictly when the muscle is particularly active. If you are a runner, you'll reach a point in your run where your muscles are no longer getting sufficient glycogen, or rather glucose, from blood. And at that moment, the glycogen in muscle begins to break down for local use. In the brain, it has been quite mysterious what the glycogen is for. The roles are emerging. We begin to see that it's great for emergency circumstances and when you're involved in intense neural activity. The peripheral nervous system, the joke is that for frankly, until about 3 or 4 years ago, there wasn't any reliable evidence that normal peripheral nerve had any glycogen. So this is a real late-breaking story. I promised a word about the relative amounts of glycogen per wet weight of tissue. Liver is clearly the champion, muscles next, but the brain has a not insignificant amount of glycogen, in the neighborhood of 5 to 10% per unit weight of what you might find in the liver. That's a fair amount. It thus was always a bit of a surprise to me when I would read in textbooks that the amount of glycogen in brain, in fact, you can find some today that still repeat this incorrect statement, is too small to have any biological significance. All right, so part one, we're going to talk about CNS glycogen and whether or not it has any benefit, if it can provide any benefit for brain. And let's propose a hypothesis. This is how you're going to write grants in the future. You're going to state a hypothesis, and you're going to have in mind some particular experiments that you'll use to prove it one way or the other. Here's ours. Astrocyte glycogen can provide fuel for neurons and axons. And I don't have time to go into the whole story. Let me give you a few facts that are compatible with this notion. The first one is that there are astrocytes every place. If we look at the central nervous system, white matter or gray matter, there are astrocytes every place, and they are in intimate contact with neurons. There's no neuron in the body, in the brain, in the peripheral nervous system, that does not have a nearby glial cell. So that's an impressive relationship, and I think we haven't spent enough time sorting out what the implications of that are. Astrocytes, but not neurons, are constantly pumping lactate into the extracellular space. This never made any sense. Why would the nervous system accumulate precious fuel like glucose and then put it into the extracellular space in substantial quantities? You'll see in just a bit how much, without it being meaningful. But for the longest time, it was an orphan fact wandering around looking for some reason. And finally, inside the blood-brain barrier, after things get past the barrier of the astrocytic end foot and the cerebral capillary endothelium, once a substance gets past, the rules are different. You've been told over and over again that your brain can only use glucose. That's a lie. Your brain is wonderful at using a great range of fuels once you're past the blood-brain barrier. If they ask the question what your brain can use for fuel before the blood-brain barrier, then it is almost exclusively glucose. But once you're past that, then it's an omnivore. It can use lots of different molecules, including monocarboxylates like lactate. So important things to bear in mind as we proceed. This is just I've always thought it kind of a telling picture. It's a slice of cortex provided by my friend Micah Nettergaard, showing you that in mammalian cortex, as in all parts of mammalian brain, there is a fairly even, maybe the ferret's got some rules of its own, but there's a fairly even distribution of astrocytes. And if we filled each of these skeleton-like astrocytes, we're just staining here with GFAP, with a dye that completely permeated the cytosol, you would see that this was almost filled the screen. The processes are incredibly dense and fine, and they are in contact with neurons and axons. So what do we know? Here's what we know, believe me, our forefathers in neuroscience that were using primitive microscopes and histologic technique always knew, as soon as they had a name for the astrocyte, that the astrocyte had some sort of a special relationship to nutritive capillaries. So we know that that's true. We know that glucose crosses the endothelium by a transport process, and that glucose can be taken up by neural elements, and it can be taken up by astrocytes and other glial cells. Then we discovered about 40 years ago that astrocytes, and astrocytes alone, contain sizable amounts of glycogen. Footnote: a friend of mine in Barcelona, Johan Genevert, has been able to show that neurons can make tiny amounts of glycogen, very small amounts. When they make larger amounts, you have a disease. The actual physiological value of the tiny amount of glycogen in neurons is yet unknown. This is not a story about that, and I think it's fair to say that the neuronal glycogen deposit has nothing to do with the things we're going to talk about today. Then we learned that astrocytes are dumping constant amounts of lactate into the extracellular space, and what we didn't know, and what we still don't know, is when that lactate has some serious physiological implications, and most especially how this all comes about. To study something in the brain, you need a preparation. And the preparation that I and my colleagues have used is the mouse optic nerve. It's badly named. When I say nerve, you might think of your sciatic nerve in the peripheral nervous system. The optic nerve connects up the retina with the rest of the brain by way of the optic nerve. So it is pure central nervous system white matter. It has oligodendrocytes, astrocytes, all the typical characteristics of white matter anywhere else in the brain. I want to make a very important point. The axon depends on its own local uptake of substrate to make ATP. It does not get any ATP from the cell body. Parts wear out, proteins wear out, enzymes wear out, and they're delivered by very slow transport to the axon. But if Daniel wants to send a message from his eye to his brain, he has to take up some glucose by those optic nerve axons, manufacture ATP to have an action potential. Okay, what else do I want to say? This is a very convenient preparation. It's easy to take out. I can take this from any animal of any age. We are somewhat hampered using brain slices because they're a lot easier to keep alive when we take them from a very young animal, and almost impossible to keep alive when we take them from a very, very old animal. That is not true from this preparation. So it's very easy to study, and it has a great functional readout, the compound action potential, and I'll show you that in just a moment. Now, when you've been listening to talks about neurobiology, no one has apologized for not telling you about white matter. Has anyone gotten up and said, I beg your forgiveness for not talking about white matter? And yet they should do that. And the reason they should do that, Monica, every single time is because when you look at the amount of white matter in a rodent, it's about 10 to maybe 15% of the entire brain. When you get to the magnificent creation that we call human Homo sapiens, it's 55%. So as we sit here, 55% of your brains is white matter, and yet it has been largely ignored. And to a very bad consequence, by ignoring it, we forget that a tremendous amount of human pathology, stroke, multiple sclerosis, many other diseases that are devastating, affect white matter and white matter only. So here's what the preparation looks like. A rodent volunteers its optic nerve. I put it in a chamber. It's the same chamber that many of you have used or seen with a brain slice floating in it. The optic nerve is unbelievably robust. I haven't had to cut any of its surface. I've just peeled off the dura, snipped the two ends, and put it in. It's still covered by the meninges. It will last, actually, I don't know how long it will last because I've never found a graduate student willing to stay beyond 24 hours next to the optic nerve, monitoring its excitability. I don't think there's anybody really jumping up and anxious to answer that question here. So I hook to one end a suction electrode and a suction electrode on the other end. This is connected to a stimulator, and I apply a maximum pulse that creates the maximum compound action potential. So I have excited every excitable axon, and I get a compound action potential. And this compound action potential moves down the optic nerve and is recorded at the other end. I quantify all of this by measuring the area under the compound action potential, which is, mathematically speaking and intuitively speaking, completely and exactly and quantitatively proportional to the number of excitable axons. So I have a wonderful, simple thing that I can use to quantify the health of these axons. All of these experiments are done at 37 degrees. Anybody who gives you a lecture about metabolism and is doing the experiments at room temperature or even at 35 degrees, you can take a nap or you can politely leave, because what you'll be listening to won't make any sense. Enzymes have very high temperature sensitivity, excitability, consumption of various metabolites varies depending on the temperature. You have to be at normal body temperature. Okay, and then you can do bad things, like remove all of the glucose or remove all of the oxygen. The data looks like this. Here's the area under the compound action potential, and I will always set that to 1, meaning that 100% of the axons are excitable under control conditions, and I'm going to call that 1. And this is very stable, as I say, for many, many hours, 18, 20, I don't really know how long because I've never tested that question, unless you do something bad, like remove all of the oxygen. And if you do that, and believe me, we didn't even know the answer to that simple question 15 years ago, the compound action potential disappears almost, well, very quickly, let's say, within 4 or 5 minutes, the amount of time for you to do a complete exchange in the chamber, and the compound action potential disappears, and then it's 0, nothing to measure for the duration of this insult. And then, depending on the duration, when you remove the insult and restore normal conditions, you get partial recovery. So I want you to think that when I've done something like this and started with 100% normal axons, I'm left after variable periods of insult with X% normal axons, in this case, 35, 40%. So now that was the wind-up. Now I'm going to show you some data. I'm skipping some steps, but you're very smart, and we have better things to do on a Friday afternoon, right? So we're going to get to work. We first started studying anoxia, and when I forget this glucose momentarily, this red line shows you what happens to the compound action potential when I take away all of the oxygen. And when I take away all of the oxygen, as you can see, the compound action potential falls very quickly to 0. I just showed you that data. One day I said, I'm sure, to a graduate student, because the graduate student said, I wonder what would happen if we took away the glucose. And I said to the graduate student, shame on me. I said, that's a stupid experiment. When you take away the glucose, the compound action potential is going to instantly disappear. And he said, I did the experiment because I forgot to put the glucose in. And then he said, but I think it's interesting. So when he forgot to put the glucose in, the compound action potential, these are minutes, ladies and gentlemen. This isn't a couple of milliseconds. For 20 minutes at 37 degrees, the compound action potential, and I was eliciting it, a couple of these, supramaximal compound action potentials every minute, just to monitor excitability. This thing looked like it didn't care at all that it didn't have any glucose. It just ran along, usually 20, 30 minutes, and then finally it would start to fall. And after about 60 minutes at 37 degrees, with no glucose whatsoever, this thing finally died and then would make a partial recovery depending on how long I had done this. So that begged the obvious question, what is this thing having for lunch? What is the optic nerve doing in the absence of glucose? Well, I started thinking about that, and I suddenly realized there's glycogen in the brain, but no one pays any attention to it. I got my friend Joel Black at Yale to do some nice electron microscopy, and these little dots, you have to make sure that you're not looking at endoplasmic reticulum, granular endoplasmic reticulum. That's easy enough to do it if you're an expert with electron microscopy. These little dots are glycogen. And you can see that there's glycogen scattered around in the optic nerve in this electron micrograph, and it is in every astrocytic process. So plenty of glycogen was there. Okay, that's interesting. So back to our hypothesis, glycogen fuels axons during zero glucose. You know, you use Latin when you want to impress your friends and the people reading your grants. So we used aglycemia, but just no sugar. No sugar, and the way this works, the way the glycogen feeds the axons is by breaking down the glycogen to lactate and transporting it to axons. If that were true, and this is what you do with a sheet of paper before you do anything in the laboratory, you make some predictions and you work with these predictions and you try to defeat them. Why it won't give you a good answer, why it gives you 3 answers instead of 1 answer. And pick the ones that give you an answer that you can't wiggle away from. So some things you might say, the axon function should decrease when the glycogen is lost. That sounds pretty unequivocal. Axons should function longer if you could increase the glycogen, right? Take away the glucose, but now you got more glycogen. If glycogen's important, then it should be able to function longer, and vice versa. If you reduce the glycogen, then it won't function as long. And if lactate's supposed to be important in your experiments, then you should try to demonstrate that there's lactate there in the extracellular space. So let's see. Here's glycogen content. This is wet weight measured by simple biochemistry, old-time biochemistry. This assay was invented 100 years ago. And here we're measuring compound action potential. This little bit of these little boxes that appear 1 a minute are what we've been looking at, the compound action potential. Here's zero glucose. This is a parallel set of experiments done on optic nerves measuring under the same conditions glycogen content. I haven't told you too much about the biochemical content of white matter, and I'm doing that now. Here's the scale, and it doesn't really mean much to you, but there's a lot of glycogen, maybe a little less than you might find in cortex, but plenty of it in white matter. And here's the punchline. When you take away the glucose, and you keep looking at the function of the optic nerve, it continues for this long period until the glycogen has gotten down to a low level. You might say, why doesn't it go to 0? It doesn't because for some reason there's a protein core around which glycogen is built, and unless you do some horrid things to mammalian tissue, there's always a bit of glycogen left around the core, even in the absence of glucose. So this looked promising. It looked like the loss of the compound action potential was exactly in temporal relationship with the reduction of glycogen. Everybody along for the ride so far? You with me? Cool. Okay, now it would be so powerful if I could manipulate glycogen content in my tissue. And it turned out to be so easy. If I take and change the bath for about 30 minutes, actually, you have to do it for an hour. If you change the bath glucose concentration for 1 hour, you can move glycogen content all over the place. So normally we bathe in 10 millimolar glucose, not because there's a state law or a federal mandate that says you have to use 10 millimolar glucose, but I can defend this. It turns out that the blood glucose in a mouse is quite high, and that's about what it is. So 10 millimolar glucose gives me this amount of glycogen. If I bathe in much lower concentrations of glucose, I think this is 2.5 millimole. You can see within an hour it drops. It's very stable at that level. I can also increase it in a very statistically significant way. And now it's time to do the experiment. So here are a series of compound action potentials that start at different glycogen concentrations. So let's take 2 millimolar, the one that was bathed in 2 millimolar glucose had dramatically reduced the amount of glycogen. Here's normal. I'm down here. This is the curve of interest. Now I take away zero glucose, and it's exactly the way that really smart Dr. Ransom predicted it would be when that graduate student approached him. No glycogen, and this drops like a rock. But the graduate student was looking at this data, right? When there's average amounts of glycogen you get here, and when you take and increase the glycogen content to way up here, now you've moved it from this inflection point at 1 minute. At 1 minute, this starts to drop to 30 minutes. So a huge effect of glycogen. So what's going on? This is a little stick diagram. I had to go into science because I have negative artistic capability. But I can do these boxes pretty well. Astrocyte here, here's an axon. Under normal conditions, you've got glucose. It can be transported. It requires a specific glucose transporter 1 into astrocytes. Axons and neurons have another glucose transporter. None of these depend on insulin, by the way. You don't need any insulin for glucose transport in the brain, the way you do in muscle and other tissues of the body. Insulin independent. That's the beauty of these particular transporters. So glucose goes in, everything is fine until you take away the glucose. Now, what the liver does is this. The liver says, all right, we'll break down some glycogen, and it gets broken down to glucose 6-phosphate. It's already phosphorylated, so that's why it's so fast, no ATP needed. And I can split this off with glucose 6-phosphatase, and now glucose can be transported out by another glucose transporter, which actually is present in astrocytes to some slight extent. And you can put glucose back in the extracellular space. Everything is fine. There's a problem because your brain doesn't have any glucose 6-phosphatase. The glucose you phosphorylate is meant to stay inside the cell, which took it up in the first place. You don't want to lose it and then have to import more glucose. So that's not going to work. And in fact, this put us in a position of thinking that the far more probable thing was that the glycogen was being broken down through anaerobic glycolysis to pyruvate, and under certain conditions, this would be shunted primarily to lactate. There are transporters, monoamine carboxylase transporter. We're not going to talk about them, but you need them because this is an anion. It gets transported here to the extracellular space, taken up by similar transporters on axonal or neuron membranes, and then converted back to pyruvate, and you can make ATP. Now, I know what you're saying. You're saying, but this is lactate. It's already been partially used. I don't want food that someone else has already partially eaten. The truth is, the step from here down to pyruvate generates almost no ATP. By the time you extract the amount it costs you to phosphorylate glucose, you get out maybe 2 ATP out of the 36 that are produced. So this is not so much about energy production. Where the energy really gets produced is in oxidative phosphorylation, the overwhelming majority of energy. And now that you know that, you realize that lactate is loaded as a fuel substrate. And if I can get it into this starved neuronal element, I'm going to be in good shape. All right. So, well, it's obvious, right? I got to see if there's any lactate around, because if there isn't, I have to get a new hypothesis. So this is an enzyme electrode, and they're very elegant things. They're a little bit big. They're not, the tip that does the measuring is small enough that you need a little magnifying glass to really see it, but it's not a microelectrode, the kinds of things you're so used to looking at. And it basically is a bunch of fancy enzyme glued onto a wire, such that when this fancy enzyme, which is interested in lactate, it's a lactate oxidase, it generates electrons that you can measure electrically. and it's specific for lactate and highly specific. So this is now commercially available. You press it against the skin of the optic nerve, and it's in equilibrium with the extracellular space. And you might be a doubter at first blush and say, boy, that seems like you're going to have bath water, contaminating your signal, and it's not going to be very fast because you said it was big and the extracellular space is small. Sometimes, in spite of your own best judgment, you need to just try it. You need to just try it. It may be an experiment that won't work, but sometimes they do. This works brilliantly. So when you measure the extracellular lactate concentration, it is on average about 0.6 millimole. That is a huge amount of substrate. It's actually higher than the typical concentration of glucose in your extracellular space. It was also amazing that it doesn't change very much when you change bath glucose concentration. You're not taking it to 0. Let's look over here first. You're going from 5 to 10 to 30. The extracellular space lactate never budges once you're above about 2 millimole. When you go to 2 millimole, it starts going down. If you go down further, it goes further down, et cetera, et cetera. So there is a design principle in our brains, in mammalian brains, to keep extracellular lactate at a high and nearly constant level all the time, except when bad things happen. And we'll come to some bad things here in just a minute. So let's do a bad thing. What we're really interested in is our hypothesis. We're wondering whether or not lactate might be a fuel for the brain under conditions where I don't have any glucose in the bath. Okay, so we can do that experiment. We got the lactate electrode. Here's our compound action potential. It's being scaled over on the right-hand side. Lactate sitting around 0.6 millimole, as we talked about. Now I go to zero glucose. I'm not going to show you the rest unless you imagine right now what lactate's going to do. Okay? You have to make a prediction. That's what scientists do. If you just go into the lab and you have no predictions, you're going to be in trouble sooner or later, mainly sooner. Okay, I'm going to predict that it's going to go down. And it goes down like a rock. Now remember, this thing is pressed against the extracellular space, so I can't measure super low levels of lactate within the tissue because I've got an equilibrium at the edge. So it's probably not a surprise that this goes down to what looks like 0. I have other data I don't have time to show you today. It's indirect, but it suggests that it goes down to a very low level and then another super low level just at this moment, when the compound action potential fails. So you would interpret that, I hope, as lactate disappearing under conditions where there's no glucose, going down to this low level, and ultimately 0 when the compound action potential no longer has any lactate substrate. I guess that's what that's all about. So it would look like this. Everything is fine with our little model until we take away glucose. Then the glucose disappears in all the places that need it, and we have to call on glycogen. We know it gets degraded because we measured that directly. We know that there's lactate in the extracellular space, and we know in the absence of glucose that that lactate goes to a very high level, and we imagine it's because it's being taken up by neural elements, in this case an axon, a central nervous system axon, and being used to keep excitability intact for a period of time. Until, of course, that lactate is gone, and then finally everything is gone. So is there any evidence that this might go on in intact tissue? My friend Ray Swanson, a very, very good scientist, and somebody, well, a very good scientist. Ray's been interested in these kinds of questions for a very long time. This is astrocytic glycogen. See this, this little histogram here that Ray measured in cultured astrocytes. When he took away glucose, the glycogen fell. Okay, we know that, we expected it. He was interested in drugs that might increase glycogen, and he found a bunch of them. But this one was special. When he put this drug on, glycogen content went very high, went way up, because it blocks a degradative enzyme. So maybe it's glycogen that can never get broken down. But the cool thing about this particular drug is when you go to zero glucose, you overcome the block. And now all of this glycogen that you've saved up for a rainy day becomes available. And it can get sucked down to a level to provide a huge amount of benefit. Ray used that drug with some anesthetized mice. And what he did was he gave these mice a terribly whopping dose of insulin to reduce all the glucose in their blood. You've heard people say, oh my goodness, I'm hypoglycemic. Bring me a jelly roll. Oh my, yeah. So that's what he did to these mice. He made them unbelievably hypoglycemic. And he had two groups. He had a group that was just normal. And when he did that, he measured how long their EEG went from this. Here's a mouse EEG. Mouse is thinking its thoughts, picturing its next cheese meal, going along being a mouse. Everything's fine. And then after I give this poor mouse all this insulin, the EEG finally goes to zero. No more cheese, no more, thinking of gnawing on your electrical wires tonight, nothing. This guy's gone. And the amount of time it took after the insulin injection was about 90 minutes. After this drug, a huge protection in a whole animal. And that's all this drug does, is change glycogen. So Ray provides us a wonderful explanation or a wonderful demonstration of the importance of glycogen in a whole animal. Now there's one other point, and I thought I could leave it out. I could have certainly left it out for our brief conversation today. But this still is over impressive. This essentially doubles the amount of time that you can survive. It takes it from 90 minutes to 3.5 hours on average that you could survive in an animal that really has a high metabolic rate in the brain. And I often, when I did the arithmetic for how much glucose or it would take, how much lactate it would take, I realized I wouldn't have predicted this much of a benefit. I started reading some older literature, and this gets back to the idea of ideas come from the combination of thinking and reading. You can't just do one. It'll get you nowhere. You have to do both. You have to read, and then you have to think about what you read. So by accident, someone gave me an old book written by, gosh, did I put the guy's name? Yes, this guy, Achachka. He died recently. He was a animal biologist interested in everything, mosquitoes, dolphins. Occasionally, he even had an interest in a human being, but mainly animals. And this guy did some brilliant biochemistry, and what he found was that during aglycemia, animals, and that includes the animals in the room here, can dramatically reduce the demands for glucose by depressing metabolism. You have built into your genes a bunch of emergency steps. Your owner's manual, it's way in the back in small print, stuff that it does automatically. When there is a catastrophe and there is no glucose, this thing slows your metabolism down. And it permits you to survive without permanent damage for much longer than you would normally have been able to do. Okay, that's great. But the real question is, what time is it? I think I have a few more minutes. Do I have a few more minutes? I think I do. Octavia, I'm having so much fun, I got carried away. Okay, anyway, so the real question that you guys have is, none of you are intending to have an episode of profound hypoglycemia anytime soon. That's not on your list of, I've got to do this, I've got to try some Don Julio tonight, I got to get severely hypoglycemic. No, but you would be more interested in knowing whether the astrocytic glycogen has any benefit while you're sitting here. So that's what we wanted to get at. And we thought and discarded about 100 experiments before we thought, there are some crappy blockers that are supposed to be specific for the uptake cell. So I get a blocker that I can give you, which won't prevent lactate from coming out, but will prevent lactate from being taken up. Now, I want you to suspend critical judgment a little bit, because these are not perfect. We've had to use 4 or 5 of these blockers to convince ourselves that we had a legitimate result. But I'm convinced of it now. Here's the experiment. So here's the compound action potential. And at this point, I'm doing something different that we haven't talked about. I'm stimulating it like crazy. I've got this electrical stimulator on the optic nerve, and now I'm making it go 100 shocks per second. Your optic nerve can follow that, because occasionally the intensity of visual images is such that you need to carry that information back to the brain. You know, we've talked about the visual system at a couple of points during our stay here in Mexico City. We didn't get into a lot of details, but the optic nerve has incredible carrying capacity. So we did this 100 shocks per second for 4 minutes. 4 minutes, very intense. And during that period of time, glucose in the bath is completely normal. You'll notice that the compound action potential actually gets larger. Well, we didn't invent new axons. There's a little bit of potassium accumulation, and it makes the electrical event higher. That's all that is. So it's an artifact. It simply means everything is fine. When I turn it off, there's a little bit of readjustment, extracellular space changes, and I come right back down to the baseline. During that period of time, there's a very significant reduction in glycogen. So even though glucose is around, during this intense sprint, I'm trying to do the 4-minute mile, right, in essence. And during that time, I'm just not going to make it, and I'm breaking down a bunch of glycogen. That's what you might say after that result, but you need to test it. So you can test it using this drug, which blocks the uptake of lactate into any kind of cell. You repeat exactly the same experiment, but in the presence of this uptake blocker. Go along. Now you do the 100 hertz for 4 minutes. The time bars are different, but it's the same period of stimulation. And rather than going up and then coming down to baseline, this actually declines. A bunch of the axons stop working. And when you turn the stimulus off, you have a reduction in the baseline. You've actually killed some axons. The danger of too much study, right? You know, you haven't had the candy bar, staying up all night studying neurophysiology, and you lost some neurons because, you know, you burnt out some of the circuits. So this would be probably inhibited normally by a bunch of control mechanisms built into your neural machinery that you wouldn't have to think about. But this is pretty impressive. At least we thought that was good evidence in favor of the idea that everything is fine until you have this massive discharge. And at that point, you run out of glucose in the neighborhood, right? Because it doesn't, transport is tough from these little capillaries all the way over here to the neural element. And at that point, glycogen comes into play, gets over here, gets back down in here. You might say, well, what about oxygen? Oxygen operates at such low levels, at the level of the electron transport train, you can't reduce it. It's still able to do oxidative phosphorylation. There was a lot of conversation about what's the trigger? How does this activity tell this neighboring cell to break down glycogen? It's a whole terrific story. Brian McVicker had a Neuron paper, I think about 2 or 3 years ago, with part of the answer is the accumulation of potassium. Just super quick, optic nerve, we're recording lactate, right? And we're stimulating, just little bursts of stimulation. Lactate goes up like crazy when you stimulate. And at first that puzzled me. You know, you're stimulating, you're probably increasing metabolic demand for these short periods of time. If you do it long enough, it starts to kind of come down. Remember, there's still glucose in the bath. But then we looked at what happens with potassium. Here I'm putting on little bits of potassium. Lactate shoots up like crazy. So this looks like a very cool engineered feedback loop. You stimulate, potassium goes up because it always does in the neighborhood of active neural elements, and it's hooked up to machinery that immediately elevates lactate in the extracellular space. We don't have time to go further with that today. I do want to point out this is a Cell paper. I'm sorry, oh yeah, here it is. A Cell paper that came out not so long ago. A number of people, the driving force is Christina Alberini, and the outcome of this study, which is thrilling, is that you can't learn properly certain things. If you're a mouse going to college here at a famous university, you can't learn properly, in this case, to stay away from a colored slab where you're going to get a nasty electrical shock. You just can't learn that, good lesson, unless you have glycogen breakdown and the availability of lactate. And now there's a dozen other examples of learning and memory being dependent on glycogen and lactate metabolism. So I'm going to summarize this very quickly and just tell you that there's something very interesting and parallel to this in the peripheral nervous system. So just to remind you, we're now looking at the peripheral nervous system. The model is sciatic nerve, everything done at 37 degrees, although your peripheral nerve has been designed to be cooler. I was sitting in the back and icicles were forming around me. It was very cool in the back. And I'm pretty sure that my peripheral nerve was not at 37 degrees for a short time, but it still worked. I could still pick up a pen and write down all the things that Daniel was saying. So here's the retina, or excuse me, here's the spinal cord, the central nervous system, and here's an axon in your sciatic nerve. No more oligodendrocytes. We're in the periphery. They're Schwann cells, but they're myelinated axons. There are also lots of unmyelinated axons. Again, the axons are on their own in terms of making energy. It looks like this. Let's look at this B panel and D panel. At fairly high power, you have these very large axons, and they're called A fibers. A fibers are always myelinated. They're associated, each node, with a Schwann cell. Here's the Schwann cell nucleus, the cytosol. Here's the beautiful, tight, dense wraps of myelin and the axonal axoplasm. Then you've got some tiny little axons. Look at the difference in size. These individual things are the C fibers. At high magnification, the C fiber is completely unmyelinated and it's cradled in a loving fashion by the Schwann cell. But the Schwann cell is not making any myelin. When you do the same kind of experiment you're used to looking at 37 degrees, stimulating electrode, recording electrode, you get a different kind of compound action potential. You get the A fiber, very fast conducting, super fast conducting, and you get this tiny little event from those unmyelinated fibers. They're tiny, they don't generate very much electricity. And then you can do bad things in the chamber, just like we've done. Here's a real recording. This is the A fiber. It's a huge electrical event. Your A fibers are meant to tell you someone just stepped on my toe. I hit my thumb with a hammer. It's getting, I'm burning my hand. Get it out of there. A fibers tell you lots of things in a big hurry. The C fiber is much slower, same time scale. And here's the A fiber. You can see how much I've shrunk it. Finally, it seems like a century later, after this guy, you get the C fiber, and it's tiny. But you can measure peak to peak height, and that's what I'll be showing you in just a moment. Is there glycogen in peripheral nerve? There's a huge amount of it. And this is, again, I'm showing you at the same time. I can adjust the amount. Here's what it is at the normal mouse glucose concentration in blood. It's much more than in the central nervous system. Who knew? Here's the rat optic nerve for comparison. So here's CNS glycogen content, your peripheral nerves are loaded with glycogen. And you can move it. You can change the concentration. It doesn't matter about that. So where is the glycogen? This is so cool, right? We looked every place. We blinded the observers. We looked at axons, Schwann cells. There were some other itinerant fibroblastic elements in peripheral nerve. Almost all the glycogen granules are in Schwann cells. But you have to be careful, as I said. Here's endoplasmic reticulum. So you need a trained electron microscopist, but all of these granules are in Schwann cells. Then you can make the same kinds of predictions. Glycogen is going to fall in parallel with axon loss when you remove glucose. Axons should function longer if you have more glycogen. Blocking glycogen will have a bad thing. Lactate should be present, blah, blah, blah. So here we go. I shouldn't do that, really. I shouldn't do that because it's my data, right? I should be excited. All right. So here's the compound action potential over here. This time, remember, I'm just doing the peak to peak for the A fiber and the C fiber completely separated, measured separately. In blue is the C fiber, and in this turquoise color is the A fiber, the fast conducting fiber. I take away the glucose, and the first thing you notice is that the C fiber falls immediately. So that was shocking. We thought we'd probably done something wrong. No, these unmyelinated, terribly important things to let you know when your hand is actually sizzling. You're at a barbecue and your hand's too close to the fire, and at first you get some heat that can be conveyed by A, but then your hand's on fire. And it's good that the C fiber tells you, get your hand out of the fire, but it's gone. As soon as you remove the glucose, very quickly it disappears. The A fiber hangs on and hangs on. Here's glycogen. It's beautiful, right? You would say, I hope, that this corresponds very nicely again to the loss of all the glycogen, whereas the A fiber is gone immediately. It just doesn't care about glycogen, or excuse me, the C fiber, these unmyelinated. Could I move glycogen content? And I'm only showing you for the myelinated, very large A fibers. Here's control conditions, the little square. 2 hours after taking away the glucose, finally, this guy starts to fall. When I got rid of glycogen, it looks like the C fiber in this peripheral nerve. And when I increase it, it goes way out. And I have to tell you, this range is mind-boggling. So depending on when you do this, you're going to lose all nerve function. Well, not all, but you're going to start to lose nerve function in a serious fashion after 20 minutes in the absence of glucose, and it goes to 3 hours. Surgeons frequently put a tourniquet if they're going to do peripheral nerve surgery or surgery on your arm, cutting off all blood flow to the tissue in your arm, including, of course, to nerves. They don't do that when they're doing surgery, general anesthetic. That is, they don't put a rope around your neck until there's no blood up there and you're quiet. It doesn't work. So the point is, this is a huge, huge range of protection. So why is it that the A fibers are so benefited and the C fibers are not benefited at all? I'm going to tell you that a very respected investigator wrote me an email about 6 months ago and said that I have to tone this down a little bit because he has some evidence that it may not be exactly as I'm telling you. But he's not here today. So if I asked you to make a hypothesis about this, I am pretty confident that one of the hypotheses would be that there is all kinds of glycogen in these A fiber Schwann cells and very little glycogen in these C fiber Schwann cells. And your PI, the lab chief, will say that is so stupid because it's just too simplistic. It's not that way. So then you do the experiment, and what you find is that it is exactly that way. There's way more glycogen in these myelinating Schwann cells than there is in the cytosol of these non-myelinating Schwann cells. And whether it's absolute, I'm willing to concede the point that I may not have this entirely correct, but certainly we did this as carefully as we could with what we had at hand, and it comes out just as we've talked. You've seen this before, except I've changed the label, Schwann cell now, and peripheral nerve axon A fiber, and we think it's like this. And in truth, I just don't have time. We've been able to measure lactate in the extracellular space, so it works very nicely. So here's what we've talked about. And I'm just going to first summarize the sciatic nerve, since that was last on your screen. It contains a lot of glycogen, and it appears to be mainly in the myelinating Schwann cells. The large myelinated axons, the A fibers, are supported by the glycogen for 2 hours under normal conditions, and it can be much more if it's increased. The glycogen clearly forms lactate, which is necessary to support the peripheral nerves. And the C fibers, for reasons that just aren't clear, appear to be on their own. When the glucose is gone, they begin to disappear. So this has been a lot of fun. It hasn't answered all questions in the field of brain energy metabolism. The field is wide open, and if your inclinations were in that direction, I would tell you that you couldn't pick a better area. Do all neurons prefer lactate over glucose? This brings people almost to the point of fighting with one another. There are some people who are so against this notion, but that, yeah, anyway, it does. And so astrocyte glycogen in learning and memory, since I made this slide about 6 months ago, there have now been 3 or 4 more papers suggesting that it's almost always the case. The signal for glycogen breakdown in the peripheral nervous system hasn't been studied, although we can measure potassium increase. We don't, there's some good possibilities. What are the implications for this? What does peripheral nerve glycogen have to say about diabetic neuropathy, where there may be big changes in glycogen availability, disease states, et cetera, et cetera. I have had so much fun. Daniel and Octavio, thank you again for inviting me, and thank you all for being here and obviously really taking part in this 3-day meeting.
Thank you very much, Bruce. ¿Preguntas?
I was a bit surprised about the glycogen and the C fibers, because, so as far as I know, the C fibers, have this wind up and this post discharge, and then they are very slow adapted, so they just fire all the time. So they are like really needs a lot of energy to function, and it's the totally opposite of A delta or A beta fiber, that they just fires once or twice. So I was expecting like C fibers would be like have more prepared for sustain this high metabolic rate. So that was a bit surprised. And then I was wondering what happened with neuropathic pain when these C fibers are changing totally and then just firing all the time.
Yeah. So there's no, you and I really would need to talk about this, and we'd have to find a factual topography that we both agreed with. So I would dispute a couple things. I think the A fibers fire very fast. They're very high frequency fires. It's like your motor neurons, right? Same classification system. They have to fire very fast so that you can do a maximum contraction. C fibers have very slow conduction. They don't follow high frequency firing. They begin to roll off pretty quickly. They have a terrible surface to volume ratio. On that point, I'm in total agreement. They have tiny amounts of volume for their amount of surface area. They're like a baby. That's why babies get cold so easily. They're all surface area. They have no volume. For C fibers, they're all surface area. They have very little volume. So a couple of action potentials, they've accumulated significant amounts of sodium. They got to get it out. So they have high energy demands. Why this design? I have no idea. Why would you not put glycogen for everybody to enjoy? I simply don't know. I don't know. I mean, you could concoct some things, and I need to do more thinking about it myself. My inclination is to think, every time I see something like this, I try to think what the evolutionary advantage might be of having C fibers that have no glycogen. Are the C fibers in charge of telling you when you should eat? It would be a heck of a lot better to have your C fibers communicating some kind of signal than to have your brain say, sorry, you didn't eat, we're checking out of here. And you've just lost 1/2 of your temporal lobe. I just don't know. And then there'd be the question of how to test this. It would be interesting to see if people with diabetes who are getting insulin exogenously, their glucose goes like this. Try as we might, even with pumps, they have episodes of hypoglycemia. Does that change things? Does it put the C fiber at risk? Like we were showing here, do they begin, does the Schwann cell have a degree of plasticity and begin to make some glycogen? Because I'll bet it has the enzymes to do that. Good questions. Can we meet later and talk about this?
Thank you for the wonderful talk. I have a question about the experiment.
An experiment done in vivo for glucose metabolism in the central nervous system? In the central nervous system. Yes and no. In brain metabolism, there are two species. There's a species like Homo ransom, okay? And then you have the people that do a nuclear magnetic resonance spectroscopy. And they measure, these short-lived radioisotope ligands that are glucose or whatever it might be. They label some stuff, and then they do systematic measurements after they do something else. They don't let you eat. They give you a little insulin. They can't hurt you, obviously, to do it at the level of a human being or a whole animal. Their experiments are extremely difficult to interpret. They're very mathematical, which I know would appeal to you, but it frightens me. I get an anxiety attack once I see their equations. And so they can say whatever they want to. I'm afraid to argue with them because they start throwing equations at me. But I don't think they've done sufficient experiments to either validate or invalidate the things that I told you about. And I rest very comfortably at night with what I've just told you about. I can tell you that at the whole animal level, people like Ray Swanson, I showed you a whole animal experiment. He's done lots of whole mice experiments, the numbers, the magnitude of glycogen, the speed of drop, all exactly the same. Now, he hasn't been able to do things as tidy as I can with this ex vivo preparation, the mouse optic nerve. But, and here you have to indulge my fantasy a little bit. For me, the mouse optic nerve is almost like having a live animal in a tiny ethical situation where I've dispatched the animal in a completely appropriate fashion. And now I have this part, which I've left exactly intact. The only thing I've cheated on is that I'm not using the blood vessels to get oxygen and glucose there. So I don't have. Yeah. So obviously it would be nice to replicate what the blood vessels do. But with the mouse optic nerve, the diffusion distances in this structure, which is about 150 microns in diameter, is such that diffusion works great. The intercapillary distance is pretty big in white matter. And so I think that the mouse, if a mouse had broken blood vessels around his or her optic nerve, I think they'd be okay with me installing a special bath arrangement and giving them oxygen and glucose in that fashion. I think their optic nerve would work perfectly fine. Adrian.
Thank you for the very nutritional talk. It's very refreshing, and you got me thinking about so many things like extracellular space. And I was just thinking about, do you know if anyone has actually looked at how the glucose moved from the bloodstream into these cells? And is it that we know that is it going through the end feet of the astrocyte, or is it somehow passing the endothelial cell into the extracellular space? And then there is something about this extracellular space that is important for. I mean, it looks like I wish to fantasize that there's something going on very important in the extracellular space. When you mentioned the effect of potassium and the exploration of design, do you know of any studies or maybe what are your ideas about?
We will fantasize later together. But for now, what I'll tell you is that, yes, there are textbooks worth of information about the territory that you outlined. How does blood get from the vascular space into the brain? So we know all the transporters, we know their KMs, we know what inhibits them, we know the rate at which they send things across. We don't know whether there are no tight junctions. So the glucose gets right across. Glucose transporter 1, a different glycosylated version of it, gets the glucose from blood into your brain. It does a great job doing that. It's driven by mass action. It goes straight into extracellular space. No problem about that. It can get around the astrocytic end feet. They're not tight junctions. We don't know whether or not quantitatively the amount of glucose taken up goes more into the end feet than it does into the extracellular space. That question is unknown. It certainly is attractive. I mean, the anatomy, how could you think otherwise, right? I mean, the thing looks like it was designed by a brilliant engineer to get the astrocyte maximum first crack at the glucose that comes across. But we just simply don't know the answer to that question. It diffuses rather quickly. Intercapillary distances are short. We think that between two capillaries, glucose probably falls almost to zero all the time because of the use. You can't, I mean, it's not a super highway. You know, you're down to little tiny capillaries. I can't be running a fire hose in that vicinity. I'll destroy the brain, which has the consistency of soft pudding. So it's a delicate business to feed you. We talk more, but I don't know if I got at any of the questions that you were asking.
Thank you, Professor, for your talk. I have a question. Experiments with lactate, did you consider of the gap junctions between the astrocytes? Lactate dynamics?
So the questions about gap junctions and astrocytes, and it's a great question. You may be aware of this paper, very nice paper that came out in Science about 3 or 4 years ago from Christian Guillaume's lab in Paris. And Christian was using hippocampal brain slices. He was using analogs of glucose that are transported but not metabolized. And then he was looking how they distribute themselves when you put them into one astrocyte and looking at neighboring astrocytes. And then he even used real glucose that is metabolizable. He took away all the glucose in the bath, and sure enough, the evoked postsynaptic response disappeared. No surprise. Now he did something that you wouldn't have expected could possibly work, and I'm sure he said to a graduate student, that is a stupid idea. But anyway, they did it. They put an electrode in that had glucose, and the glucose dripped just through the micropipette tip into a single astrocyte, and they got back a substantial amount of the evoked response a good distance away from that astrocyte. So it had to move by way of gap junctions. So yes, I'm sure that they play a role. The things we were talking about are global. I took away all the glucose, not just in one little area. If you were thinking deep thoughts just in your septal nucleus, then that area might get starved, and this pipeline by way of astrocytic gap junctions might help with focal decrements in glucose.
One very short. Have you studied whether the glycogen lactate shuttle takes the part in the preconditioning protection against brain ischemia?
Yeah, so that's a good question. So the question's about ischemic preconditioning. It turns out that the best thing that can happen to you if you're going to have a stroke is for you to have a little mini episode of vascular occlusion 72 hours earlier. And if you do that and you recover completely from it, if I close that vessel off 3 days later, 72 hours later, you're going to do much, much better. We call that ischemic preconditioning. And right now, the belief is that this is probably more a consequence of a bunch of fancy signaling through interferon 1 and through toll-like receptor 4, much more than through glycogen. So it's moved beyond my simple way of thinking. Could it play a role? Why not? I actually think that it could. And I sometimes wonder if people that exercise a particular area of their brain, and here we're still learning about this, but maybe somebody who has an especially sharp eye at the pool table can always make the shots that you can't make, and you say, I'm the same size. He's clumsy most of the time. How can he always put those balls in? He's practiced and practiced, Bushy, practiced and practiced and practiced, and maybe in an area of their brain, they just simply have higher processing power because they have more, they've built up glycogen stores. Here I'm completely talking. So I would like to ask the audience to give themselves a round of applause. I think you've been very involved. That was certainly what Daniel and Octavio had in mind, and they would have been terribly disappointed if you hadn't been involved, asking questions and paying attention. So congratulations, you made these two guys very happy.
Agradecemos su presencia y a nombre de Daniel, mío, del posgrado de Neurobiología, les damos las gracias por haber asistido. Les recordamos que las personas que participaron, que tienen acceso a constancia, se les enviará por medio de correo electrónico. Entonces solamente las personas que firmaron su asistencia tendrán derecho a esta constancia. Pues muchas gracias y esperamos que esto pues sirva para que ustedes piensen seriamente en hacer la maestría en neurobiología. Muchísimas gracias. Thank you very much.
Yeah.
Excellent question.
Yeah.
No problem. Yeah.